When we were talking about how a muscle contracts, I started by saying that we have to have a signal from a neuron that's going to cause the release of calcium. But there's a lot going on for that to happen. In order to get the signal from the neuron to release the calcium, we have to excite the muscle cell. We've talked very briefly about excitable cells before. An excitable cell is a cell that changes its membrane potential in response to some sort of signal. In this case, the signal is a signal from a neuron, and we're going to change the membrane potential of the muscle cell. Recall that the membrane potential of a cell is essentially the charge of the cell, or more specifically, the difference between the charge inside the membrane and the charge outside the membrane. This membrane potential in our cells is due to several different factors. The first important factor that contributes to the membrane potential of our cells is the sodium-potassium ATPase pump. Remember that the sodium-potassium ATPase pump is found in almost all of your cells, and its entire job is to kick sodium out of the cell and bring potassium into the cell. Each time the sodium-potassium pump works, it moves 3 sodiums out of the cell and brings 2 potassiums into the cell. Now let's think about that for a minute. 3 sodiums with their positive charges are going out, and 2 potassiums with their positive charges are coming back in. Every time the sodium-potassium ATPase works, we're moving more positives to the outside of the cell and fewer positives back into the cell. This is going to create a situation where we have more positive charges outside the cell and fewer positive charges inside the cell. The second factor we have at play here is that your cells have some potassium channels that are a little bit leaky. What that means is that they're always letting potassium run through. Keep in mind that because of the sodium-potassium ATPase pump, we have tons of potassium inside the cell and very little potassium outside the cell. We learned early on that molecules want to move from where they're more concentrated to where they're less concentrated. So the potassium wants to move from the inside of the cell where it's very concentrated to the outside of the cell where it's less concentrated. Potassiums have a positive charge, so as these potassiums are moving to the outside of the cell through the leaky channels, positive charges are moving from the inside to the outside. That gives us even more positives outside the cell and even fewer positives inside the cell. We can add to that one more big factor that plays a role here, and that is that we have a lot of very large negatively charged molecules that are essentially trapped inside our cells because they're too big to get out. There are a lot of proteins with negative charges that are trapped inside your cells, and also RNA and DNA is inside your cells with a lot of negative charges. When we put all of these things together, the outside of the cell is quite positive when compared to the inside of the cell. We say that the membrane potential of the cell is negative because the inside of the cell has a negative charge compared to the outside of the cell. And as I mentioned in our histology unit, we can actually measure this. We can put an electrode inside a cell and an electrode outside a cell and compare the electrical charge and see that the inside of a cell is negative. The membrane potential, or the charge, of a resting muscle cell is about negative 90 millivolts. So that's a fairly negative charge. When a muscle cell gets a signal from a neuron, that changes the membrane potential of the cell. That's what we call exciting that muscle cell. Let's take a closer look at the structure where this is going to happen. Let's take a look at exactly how a signal from a neuron is able to change the charge of our muscle cell. To see how the signal from the neuron can change the charge of the muscle cell, we need to start by looking at the neuromuscular junction. The neuromuscular junction is where the neuron meets the muscle. Because this is a connection that involves a neuron, we call it a synapse. Connections involving neurons are called synapses. And this synapse, or this connection, is going to be between the axon terminal of a motor neuron and the sarcolemma, or the plasma membrane, of the muscle fiber. In this figure, we can see the neuron coming here to the muscle. And when we zoom in a little bit, we can see the axon terminal, the very end of that axon here. And we can see where it's interacting with the sarcolemma of the muscle fiber. The sarcolemma in this area is called the motor end plate. And you can see that it's not just a flat area of membrane, but it has a whole lot of folds in it. We've talked about this phenomenon before, about why we want to have a lot of folds in membrane. Hopefully you remembered that it's about surface area. When we have more folds in the membrane, it gives us more surface area there. And in this case, the surface area is important because it gives us more room for receptors. And that's what we have a lot of on the motor end plate. We have receptors. These are receptors for neurotransmitter. If we go back to the neuron end of this connection, here's our axon terminal. And you can see a lot of vesicles inside the end of this axon. These vesicles contain the neurotransmitter. When a neuron fires or sends its signal, it releases neurotransmitters. So here's our neurotransmitter. The neurotransmitter is going to get released into this space between the neuron and the motor end plate called the synaptic cleft. It's a little space between them. And the neurotransmitter diffuses across the synaptic cleft and then binds to all of the receptors here on the motor end plate of the muscle fiber. When we're talking about the contraction of skeletal muscle, the neurotransmitter involved is acetylcholine. So the neurotransmitter inside these vesicles is the molecule acetylcholine, and the receptors on the motor end plate are acetylcholine receptors. The important thing about these acetylcholine receptors is they are actually sodium channels. They're sodium channels that are usually closed, but then when acetylcholine binds to them, they're ligand gated. When acetylcholine binds to these sodium channels, the sodium channels open and sodium can run through. Let's think for a minute about what's going to happen. Because of the sodium-potassium ATPase pump, we have lots of sodium on the outside of our muscle fiber and very little sodium on the inside of the muscle fiber. So when acetylcholine binds to the acetylcholine receptor and it opens a sodium channel, which way is the sodium going to go? Is it going to run into the cell or is it going to run out of the cell? It's going to come in. Lots of sodium on the outside, very little inside. Sodium rushes into the cell. What's the charge on sodium? Positive. When all of these positive charges are rushing into the cell, what is that going to do to the membrane potential right there? What's going to happen to the membrane potential of our neuron right there where the acetylcholine receptors are? It's going to become more positive. We're actually going to increase the membrane potential. The inside of the cell is going to get a little positive because of all the sodiums rushing into the cell at that point. Now this isn't going to have an effect across the entire muscle fiber yet, but just right there where our acetylcholine bound to the acetylcholine receptors, sodium comes in and increases the membrane potential. In order to make this a bigger event, in order to actually excite the whole muscle fiber, we have to have a different type of sodium channel nearby. And nearby to those acetylcholine receptors, we have voltage-gated sodium channels. These are sodium channels that are usually closed when the membrane potential is negative, but when we change the charge of the cell and the membrane potential becomes positive, they open. So we had a little bit of sodium coming in through those opened acetylcholine receptors. That increases the membrane potential. That opens the voltage-gated sodium channels nearby. Sodium then rushes in through those voltage-gated channels, which raises the membrane potential in that area and opens more voltage-gated channels, which lets more sodium in, which raises the membrane potential more, which opens more channels. Do you see where we've got a positive feedback loop going on here? More sodium comes in, we open more channels, more sodium comes in, we open more channels, and this spreads across the entire sarcolemma. So the entire muscle cell now is excited. We've changed the membrane potential of the entire cell. That big change in the membrane potential that spreads all the way along the muscle fiber is called the action potential. We see action potentials in muscles and we see action potentials in neurons. It's a change in the membrane potential that spreads all the way along the cell. In order to see how we get from the action potential to the actual contraction of the muscle, we have what's called excitation-contraction coupling. We have an excited muscle fiber that had an action potential, and now we have to see how that's going to cause the actual contraction. What happens is that the action potential spreads all the way along the membrane of the muscle fiber, and that includes going down into the T-tubules. So remember that the T-tubules were these infoldings of the membrane that reach deep down into each muscle fiber. And these T-tubules are associated with the sarcoplasmic reticulum. The T-tubule contains voltage-gated calcium channels, and as this change in membrane potential goes down the T-tubule, it opens the voltage-gated calcium channels. When the voltage-gated calcium channels open, they're attached to some calcium channels in the sarcoplasmic reticulum called the ryanodine receptors. And when the voltage-gated calcium channels open, they pull the ryanodine receptors open, and that opens calcium channels in the sarcoplasmic reticulum, where all the calcium is being stored. That calcium then rushes out of the sarcoplasmic reticulum and binds to troponin, which shifts the tropomyosin out of the way and uncovers the active sites of actin so that muscle contraction can begin. That's how we see the coupling or the joining of the change in membrane potential, the excitation, and the actual contraction. Let's see how this whole thing fits together from beginning to end. We start with a neuron that releases acetylcholine into the synaptic cleft at a neuromuscular junction. That acetylcholine binds to acetylcholine receptors in the motor end plate of the muscle fiber. Those receptors are sodium channels, and when acetylcholine binds to them, they open. Sodium comes in through those channels and changes the membrane potential right at the neuromuscular junction. That causes the opening of voltage-gated sodium channels nearby, which lets more sodium in, which opens more voltage-gated sodium channels, which lets more sodium in, which opens more channels, and this is the action potential, a change in membrane potential that spreads across the whole muscle fiber. Now our muscle fiber is excited. This action potential spreads down the T-tubules, and it opens calcium channels in the T-tubules, which then pull open the ryanodine receptor calcium channels in the sarcoplasmic reticulum. That's what lets the calcium out of the sarcoplasmic reticulum, where it binds to troponin, shifts the tropomyosin out of the way, and uncovers the active sites of actin. Now that our active sites of actin are uncovered, our myosin can grab, pull, release, reach, grab, pull, release, reach, over and over again, shortening the muscle. And that continues as long as the neuron is still signaling or as long as there's still ATP available. In order to stop the contraction, the neuron has to stop releasing acetylcholine. Then we have to get rid of all the extra acetylcholine that's still left there in the synaptic cleft. Thankfully, we have a handy little enzyme that does that for us. The enzyme acetylcholine esterase breaks down any acetylcholine left in the synaptic cleft so that the neuron stops signaling. While this is going on, our sodium-potassium pumps are helping us to reset the membrane potential of the cell so that we can close all those voltage-gated channels. We close all the voltage-gated sodium channels, we close the voltage-gated calcium channels, and now our calcium pumps go to work taking the calcium out of the muscle cytoplasm and putting it back into the sarcoplasmic reticulum. As we get the calcium put away, it gets released from the troponin shifts the tropomyosin back in place, we cover the active sites of actin, myosin can't bind anymore, and now titin can allow the sarcomere to be pushed back out to its original length, and the contraction is over.